So it's with great pleasure that I introduce the next speaker of our session, Dr. Francois Aboud, who is Professor Emeritus at the University of Iowa. Dr. Aboud will be speaking to us today about the brain and the immune system. Dr. Aboud.
That's good. Thank you. Thank you very much. And this is, let's see here, the pointer. It's going to be very hard to see that pointer, but that's, so I'm deep privileged to be here, and I'm very pleased to see you coming to listen to what I have to say. But Kathy introduced me as emeritus. I don't know whether you have any connection with the University of Iowa, whether they made me emeritus. I am not emeritus, but I'll be getting there soon. Anyhow, so Professor Dampney presented a fantastic portrayal of what the autonomic nervous system does to maintain your homeostasis in your body. He covered an enormous array of reflexes by which the autonomic system can regulate almost all your functions. What I'll be talking about is the autonomic system as a regulator of pathologic states rather than the normal physiologic states, and tell you that it is a very powerful participant and operator in the development of cardiovascular pathology. And I'm going to say that the way the autonomic system does that is not necessarily with a direct action on the cardiovascular hemodynamics system. It's through the immune system. So the autonomic regulation of the immune system makes it a major contributor to pathologic states. So I start out by identifying some of the early work. The Nobel Prize was given to Cornel Hyman in 1938 for identifying these carotid sinus nerves, tiny nerves in the neck in here that have a powerful regulation through activation of autonomic reflexes on the sympathetic and parasympathetic output. And the effects of these baroreceptor nerve and chemoreceptor nerves in the carotid sinus is, on the one hand, to prevent any surges in your blood pressure from causing a stroke by inhibiting sympathetic drive. And if your arterial pressure would fall, the baroreflex unloading, so to speak, of the baroreceptor will increase sympathetic activity. So it took 30 years until the efferent part and the neurotransmitters have been identified by Katz, von Ohl, and Axelrod with acetylcholine as the transmitter for parasympathetic nerves and norepinephrine as a transmitter for sympathetic nerves. And they identified the storage, the release, and the degradation of the nerves. Now, what I would like to do is first give you an example of what the sympathetic nerve activity can do to you. I give you an example where the sympathetic nerve activity is suppressed dramatically, and I'll give you another example of when your sympathetic activity is enhanced chronically. Now, this is the example of what happens to you. Let me see here. If your sympathetic activity, okay, you're gonna, all right. So these people in here are in a very stressful situation, and they keep suddenly falling. And the environment is stressful in some way, but not as Roger Dampney has suggested. Maybe a lot of anxiety can do that. But you hear people laughing, and the reason that they're laughing is that because this is not necessarily fatal at all, and within a few minutes, like this guy, he's smiling maybe, or All right, here is another one. Watch that. So what I'm showing in here is a sudden loss of motor tone, and that kind of brings you down. And the fortunate thing is that this doesn't last. This is extremely common. I bet more than 2/3 of you have had an experience almost like that. So if you, maybe I can. You want to see this? Okay. All right. So, okay. Well, so I'm going to show you here in a minute what is actually happening to your autonomic system if you were like that. But the important thing is that after this fellow will fall, he's going to get up again and go out smiling. And that's really what's important. So if you were to be able to measure sympathetic nerve activity, you put an electrode in your peroneal nerve there, and just before the vasovagal syncope, the sympathetic activity is increased dramatically, and then all of a sudden it stops. And with it, watch the heart rate. There is a bradycardia, sympathetic withdrawal and bradycardia. The arterial pressure falls, and the plethysmograph on the finger shows vasodilatation. So we have bradycardia, vasodilatation, and severe hypotension. But I just told you that the baroreceptor nerve, when your pressure falls, should trigger an increase in sympathetic activity. Here the pressure is falling, and there is an inhibition of sympathetic activity. And I want to remind you that besides the baroreceptor regulation of sympathetic outflow and parasympathetic outflow, there are cardiac afferents, vagal afferents. So your vagus is not simply an efferent system, it's a sensory system, and the sensory nerve ending of the vagus in the heart can be inhibitory. So besides the baroreceptor mechanical activation that decreases sympathetic drive, the cardiac afferent, vagal afferent, can inhibit sympathetic drive and accentuate parasympathetic drive and cause sympathetic inhibition in bradycardia. Now, this second example that I'll show you is an example in which sympathetic activity is enormously increased. Now, way back, several years ago, Jay Cohn reported some data in patients in severe heart failure. And what he was able to report is that if the circulating blood levels of norepinephrine were over 800 picogram per milliliter, the survival, or less than 10% of the patients survived for 2 years. And when the circulating levels of norepinephrine were less than 400 picogram, more than 50% survived for 4 or 5 years. So this is a very powerful regulation by the sympathetic nervous system of mortality in patients in heart failure. Now, that increase in norepinephrine is caused by, here we're showing increase in sympathetic nerve activity. From top down, you have sympathetic nerve activity, and I guess in normal individuals, and then in heart failure, very mild heart failure, modest heart failure, and very advanced heart failure. And the correlation of increase in sympathetic nerve activity that causes all that circulating norepinephrine is very well correlated with the increase in end diastolic pressure, which is an indication of increasing heart failure. So clearly the baroreflex is not preventing this sympathetic activity from increasing. So the question is, can we, if sympathetic activity is bad in heart failure, causing that mortality, can we inhibit the sympathetic activity? And for a long time, it was thought that to try to cause an inhibition of sympathetic activity electrically does not seem to be effective in sustaining the inhibition. That if you put an electrode on the baroreceptor nerve, for example, and you stimulate it, you can inhibit sympathetic activity, but it doesn't last very long, until actually Lohmeyer here a few years ago demonstrated that that's not necessarily true, that stimulating the carotid sinus nerve can cause, in conscious dog, bradycardia, and hypotension. It lasts for about a week. So that raised the hopes that maybe you can modulate this excessive sympathetic drive in patients over a prolonged period of time, and the sympathetic can be still inhibited. Now, these two experiments I chose in animal models of heart failure to really demonstrate that you can manipulate the autonomic system in such a way that you can improve survival. Now, to the left is a study in Zucker's lab where heart failure was produced in dogs by pacing, and what you see below are plasma levels of norepinephrine. The main point is if you do not stimulate the baroreceptors, carotid sinus nerve, the mortality is very high. You know, within just about 40 days, you see almost total mortality, and the norepinephrine levels are very high. If you stimulate the carotid sinus nerve, the increased survival is very evident, associated with a suppression of norepinephrine level. And another model on the right is a rat model in Sunagawa's lab of myocardial infarction. Here is the same thing again. The same thing again, the survival during vagal stimulation. And the period of vagal stimulation here between 0 and 40 days is short. But the effect, the protective effect is sustained over 2 or 3, 4 months. And associated with a reduction in norepinephrine level during vagal stimulation. So two things here we're showing. In one model of heart failure, baroreceptor stimulation inhibits the sympathetic drive and probably also activates the vagus. And on the right is direct vagal stimulation that prolongs the survival. And so basically then what I'm saying is that if you have an increase in sympathetic activity and a decrease in parasympathetic activity, that's a bad actor really. And what you try to do is inhibit the sympathetic and enhance the parasympathetic. And can it be reversed in human? So there are currently trials in patients where carotid sinus nerve stimulation is being attempted in drug-resistant hypertension and vagal nerve stimulation. We're participating in a study like that in humans with heart failure may improve survival. And this is just simply an example. Now, how does that happen? Why is the autonomic system so powerful in altering the cardiovascular pathology? It's through, I would like to suggest, a conspiracy between the autonomic system and the immune system. By the way, I'm not an immunologist, but I learned quite a bit with the help of a lot of immunologists. So I'm representing this autonomic immune interaction by this triangle. The angle on top tells you that autonomic regulation increases mortality, and I tried to show you that. Angle B, I will show you that the inflammatory response is an integral part of almost all cardiovascular manifestation of disease. And then angle C, I will show you how the autonomic system can modulate the immune system in such a way that it can be increase mortality or it can be beneficial. So first, very quickly here, how the immune system contributes extensively to atherosclerosis, the plaques, and foam cells, and T cells, and dendritic cells in the atherosclerotic plaque. In heart failure, the cytokine storm causes a significant central increase in sympathetic drive in heart failure. Atrial fibrillation, invasion of the atria, actually, with macrophages and monocytes can induce fibrosis and remodeling. And stroke, macrophage extravasation causes an increase in peroxyredoxin release in the brain that can accelerate the damage process. And I'll be focusing on hypertension. And I will be talking a bit about the modulation of the two components of the immune system. First, the innate immune component, which includes the monocytes, macrophages, and dendritic cells that present the antigen, so to speak, to the T lymphocyte and the adaptive immune system. I'll be talking about the effect on both these systems in hypertension. First, the fact that removing the thymus in these nude mice prevents the onset of docosal hypertension. So you can't get docosal hypertension if you don't have a thymus. And even in spontaneously hypertensive rats, if you, the spontaneously hypertensive rat does not become hypertensive until, say, the 6th week or so after they're born. Thymic transplant from Wistar-Kyoto normotensive rat can really delay the onset of hypertension in SHR. And this is a very important experiment by Harrison's group in which he showed that now another type of hypertension, angiotensin hypertension, induces a significant infiltration of a vascular system with T lymphocytes, and that if you expose a rag knockout, the rag mouse doesn't have T or B lymphocytes, and you give him angiotensin, this angiotensin pressor response that you see here in red is not seen if the rat doesn't have, if the mouse doesn't have T lymphocytes or B lymphocytes. But then when you adoptively transfer that rag knockout with T lymphocytes, they become hypertensive, showing very clearly that the angiotensin hypertension was very dependent on whether there are T lymphocytes, and when you give T lymphocytes, you can restore it. Now the question is, how does that happen? What is the mediator? Interleukin-6 is a very potent inflammatory cytokine, and there are animals that do not have IL-6. The IL-6 knockout animal, and you see the IL-6 knockout animal doesn't have hypertension. And the first maybe inclination you may have is to say, well, IL-6 is important for the vasoconstrictor effect of hypertension, but here it was shown that the IL-6 vasoconstrictor effect on these afferent renal arteriole, in the IL-6 knockout, the vasoconstrictor effect is still maintained. So there is another component in that hypertension. And we think that the component of hypertension that is interrupted by IL-6 is probably the central activation of sympathetic activity. That suggests that there has to be a cytokine release in the central nervous system in the pro-inflammatory area that drives sympathetic activity. But then the other indication that IL-6 may be important, not just in mediating the central drive, is important apparently in the phosphorylation of JAK2 and STAT3 in the kidney by angiotensin. And this experiment clearly shows that IL-6 knockout do not phosphorylate JAK and STAT, and may have prevented the sodium reabsorption that has been ascribed to activating these genes in the kidney. Now, the direct link between sympathetic activity in the immune system is very evident from these kinds of experiments. Now, here is angiotensin giving in the central nervous system, and to your left there is splenic sympathetic nerve activity and renal sympathetic nerve activity. And to the right there is the expression, a gene expression of IL-1 beta, IL-2, IL-6, IL-16, TGF-beta. In gray is the effect of angiotensin. But in red is the response that has been partially abrogated, suppressed by denervation of the spleen. The denervation of the kidney is still intact. You denervate the spleen, and that inflammatory response in gene expression in the spleen is reduced. So that suggests that direct link I'm talking about between sympathetic activity and the activation of the immune system in the spleen. Another activation of the sympathetic system that is important in the regulation of the immune system is the sympathetic innervation of the bone marrow. And in this experiment, is portrayed in here the myocardial infarction model. So myocardial infarction increases sympathetic nerve activity. The sympathetic nerve activity innervating the bone marrow causes the release of progenitor cells, hematopoietic endothelial stem cell progenitor, to the spleen, where monocyte production is accelerated and then migrate to the endothelium of the coronary vessel and cause a vicious cycle, so to speak, additional damage to the endothelium and potential myocardial infarction. This is a clearer indication of what's happening at the level of the bone marrow. Activation of beta-3 receptor increase the migration, intravascular migration of monocyte and hematopoietic stem cells, which then go to the brain, and you get monocytopoiesis, and macrophages again end up in the endothelium of the coronaries and magnifying the thrombotic effect. So sympathetic innervation of the bone marrow can, by accelerating the migration to the periphery of these monocytes and macrophages, can induce an inflammatory response that can cause vascular damage. Another link between the sympathetic nerve activity and the transendothelial movement of monocytes and macrophages is demonstrated in this experiment. I like it. I just like it very much because it just shows that the natural fluctuation of sympathetic activity can exert a very significant effect on the transendothelial migration of monocytes and macrophages in, say, vascular tissues of any organ. So to the left here is sympathetic activity is minimal, the two central panels in here, and the migration, the transendothelial migration into peripheral muscle, for example, skeletal muscle, or into the marrow, for example, transendothelial migration into the bone marrow if you carry out a bone marrow transplant, for example. That these are exaggerated when sympathetic drive to either the vasculature of the muscle or the vasculature of the marrow is enhanced. And that could be because of activation of adhesion molecules that will facilitate the movement of these monocytes macrophages. In the muscle, it increases inflammatory responses. And in the bone marrow, it could improve bone marrow grafting. And there have been some experiments, very interesting by that group, that show that the grafting of bone marrow is more effective if it's done at night or if it's done during the infusion of isoproterenol. And that's a very, very interesting possibility, at least demonstrated experimentally. I would love to see that, of course, in humans. So I just showed you so far the effect of sympathetic activity on the immune system. Now, the parasympathetic nerve activity on the immune system is even more intriguing. So this experiment, the vagi, subdiaphragmatic vagi, were cut. And here, what you see, by photon emission tomography, the expression of NF-kappa B is markedly increased around the gut in the colon after 4 weeks of the denervation. And here in that same animal, taking lymphocytes from the spleen of animals that have had a vagotomy, it shows how those lymphocytes are very pro-inflammatory in terms of interferon gamma, TNF-alpha, and IL-6 when those cells are activated. So there is, what we say here is that your parasympathetic system, just as you're sitting here now, is controlling your inflammatory response. Because at least in these mice, when the vagi are cut, there is a spontaneous increase in the expression of NF-kappa B in the gut, and certainly there is a major inflammatory system in the gut. So that was very, very impressive demonstration of how the vagi can do it. Another very intriguing experiment was done by Kevin Tracy some years back now. And here, Kevin Tracy and his colleagues are stimulating the vagi. In what animal? An animal that received LPS. Now, LPS causes septic shock, and it does that by activating toll receptors on immune cells and increases the release of TNF. If you give LPS to an animal, the blood pressure drops, as you see here on the right. But if you give LPS after cutting the vagi, the drop in pressure is much more dramatic. And if you give LPS while you're stimulating the vagi, the hypotension is really significantly less. And the idea there is that the vagi that innervate the splanchnic bed and most of your splanchnic organs will modulate the inflammatory response of these inflammatory cells, will tend to suppress them and be protective even against the stimulation of toll-like receptors by LPS. Now, what they went on to show that this modulation of the inflammatory response in macrophages to the left there is because those macrophages have cholinergic receptor on them. And they did a lot of experiments to show that the alpha-7 nicotinic receptor that is activated during vagal stimulation by acetylcholine is an anti-inflammatory receptor. So activation on those macrophages here on the left, activation of these cholinergic alpha-7 receptors can suppress the inflammatory response and be protective. So that experiment is an LPS induction of sepsis. Now, how is that relevant necessarily to hypertension? So this is another experiment in which hypertension was produced by the 2-kidney, 1-clip hypertension. And that 2-kidney, 1-clip hypertension was induced in a knockout of the alpha-7 nicotinic receptor. And what you see in here, when the alpha-7 nicotinic receptor is knocked out, the 2-kidney, 1-clip hypertension is associated in red in here with greater increases in IL-6, IL-1 beta, and TNF-alpha. And the contractility, the changes in hypertrophy, presumably into 2-kidney, 1-clip, is enhanced in the absence of alpha-7, and the degree of glomerular sclerosis is worsened in the alpha. So here is, to your right here, the panels on the right are the alpha-7 nicotinic receptor knockouts. And it shows that there is a lot of fibrosis in the heart, and a lot of glomerular sclerosis in the kidney at the bottom there in the alpha-7 knockout compared to the sham alpha-7 knockout. But you have to induce an inflammatory or a stressor response. Now here, the stress is 2-kidney, 1-clip hypertension. And the damage of the 2-kidney, 1-clip hypertension is accentuated in the absence of alpha-7. So with that as a background, norepinephrine is bad, parasympathetic is good, and there is significant regulation of the autonomic system or of the immune system by the sympathetic and parasympathetic. We asked the question of whether the spontaneously hypertensive rat develops hypertension after birth. Do they exhibit immunologic defects? Is there an intrinsic abnormality in the immune cells of the SHR? Because I showed you already that the SHR pressure is dependent on the thymus. But here we're asking the question of whether those innate immune cells in the SHR, just after they're born, before they develop hypertension, are they normal or abnormal? How do they act? And do they react with an inflammatory response, say, to activation of toll receptor? These are the toll receptor that can sense endogenous protein or exogenous protein and initiate the inflammatory response. And is there evidence that the response of those cells cultured in isolation that they respond differently to angiotensin or to the cholinergic transmitter nicotine. So basically then the framework I'm suggesting here is a big one. I mean, suggesting here that sympathetic activity and angiotensin-induced sympathetic activity is bad, parasympathetic activity is good, and these two autonomic outputs regulate immune cells, innate immune cells that may have these receptors or actually have these receptors on them that modulate the release of cytokines, which in turn activate the adaptive immune cells, the T lymphocytes, and the cytokines and the migration of the cells themselves to the peripheral organs, the brain, the vascular system, the heart, and the kidney can induce these end organ damage. And so to answer that question, we took these immune cells, and they have Toll receptors on them, and we can activate them with Toll receptor ligand to release cytokine. And in the middle here, I'm showing that that signaling pathway, inflammatory signal pathway, can be modulated in a very effective way by the nicotinic cholinergic receptor or the angiotensin receptor to modify the amount of cytokine release, and that in turn then modifies the T cell activation and the invasion of the tissues and the development of hypertension. So again, a summary of what's going on in here. To the left in here are the Toll receptors on an immune cells. They're activated by a multitude of exogenous ligands, the pathogen molecular patterns of fungi, bacteria, or viruses, and the endogenous ligands, which are much more relevant to autoimmune system, or in that myocardial infarction model with a tissue damage that releases DNA particles. With those DNA particles and the product of cell destruction can function as ligand, and they do function as ligands, endogenous ligands of the T cell of the Toll-like receptor to induce NF-kappa B and the expression of cytokines. And then post-transcription migration or generation of cytokine may be modulated by angiotensin receptors or acetylcholine receptors. So that's the framework that we're trying to develop here. So what we did was take spleens out of WKY and SHR and isolated the splenocytes and exposed the splenocytes to ligands, the Toll receptor ligand, and those ligands are very specific. There are several Toll receptors, and each Toll receptor has some very specific ligand, and we increase the release of cytokines in vitro and see then if adding nicotine will alter that increase in cytokine, or angiotensin will alter that increase in cytokine. So what you have here from the left to the right are 3 doses of several Toll receptor ligand, Toll receptor 2 ligand, 3 ligand, LPS is Toll receptor 4, flagellin 5, clonine 7 activate Toll receptor 7, 8, and CPG activate Toll receptor 9. And here is the release of TNF alpha, IL-10, and IL-6. And what you see are 2 things. There is a dose-response relationship. There is tremendous variability of the amount of cytokine release depending on which toll you're activating. And then to the right here at the end, angiotensin by itself doesn't do very much, and nicotine by itself doesn't do very much. And there is no difference in the response of the WKY or the SHR in terms of the effect of tolls. So what are we talking about? We're talking about something different here. We're talking about, up to the left there on top, when you activate the toll receptors 7, 8, and 9 with clonine-7 and CPG, the white bar shows the release of cytokines, and the black bar shows how that release is reduced significantly by nicotine. So nicotine has an anti-inflammatory effect in vitro on the release of cytokines. Now, the SHR, in the pre-hypertensive stage, their splenocytes show a significant acceleration and enhancement of the cytokine release. So the inflammatory effect is very evident. With the same nicotine in the WKY, there is anti-inflammatory effect. In the SHR, there is inflammatory effect. With angiotensin in the WKY, we didn't see any effect. There is no suppression. But in the SHR, there is still an exaggerated inflammatory response. Summary here, nicotine and angiotensin in the splenocytes of the SHR, young, before they develop hypertension, is already pro-inflammatory. So the mechanism of that are going to be difficult to understand, but we're working on that. Now, here we try to see whether that in vitro effect in the isolated splenocyte can be reproduced in vivo. So what do? We infuse nicotine, say, for 24 hours, and at the end of the 24 hours, just about 4 hours before ending the experiment, we inject toll-like receptor ligands. In this situation, I think we invested CLO96, the toll-like receptor activator of 7 and 8, and we collect sera and tissues from these animals. And here is what we found. The in vivo serum levels of activation by CLO97 on your left there in WKY, nicotine alone in the vivo now can increase IL-6 and IL-1 beta. And the toll-like ligand, CLO97, also can increase the cytokine release. But when you add nicotine and CLO97 together, nicotine seems to reduce dramatically the inflammatory response. So nicotine has an anti-inflammatory effect in vivo. And then on the right in here, conversely, in the SHR, adding nicotine to the CLO97 toll-like receptor activation enhanced the response. So we interpreted this as meaning that those immune cells of the SHR have lost the suppressing effect, say, of nicotine that is seen in the WKY. Now it's an inflammatory response. The other things that we also notice that not only the responsiveness of those cells is increased, but also the number of cells. So here is cell sorting with a marker of CD161, and I'll tell you a little bit more about that CD161 marker, and then and CD8. And what you find that in the SHR, the CD8, CD161 positive cells are significantly increased in the SHR. You don't see that in the WKY. CD161 positive and CD8 positive. And nicotine seems to enhance that response. So we have also, in addition to an abnormal sensitivity of those immune cells, there is an accelerated production by nicotine, if you will, of inflammatory immune cells. I guess that's the summary here. Again, sympathetic activity is bad, parasympathetic is good. You can modulate the immune system, both centrally in the marrow and in the spleen. The innate immune cells themselves have abnormalities of the receptors that modulate their inflammatory responses. In the SHR, in a pro-inflammatory way, there are more of these inflammatory cells. There is a conversion of T lymphocytes to Th17 T lymphocytes, and certainly the migration to the end organ is bad. So what I have here again with your patients again, autonomic nervous system is a powerful modulator. It causes pro-inflammatory, morbid cardiovascular consequences when the sympathetic is excessive. Vagus nerve, surprisingly, provides a very protective anti-inflammatory effect that seems to be mediated through alpha-7 nicotinic receptor. Long story here that needs to be pursued extensively. And then in a genetic model of hypertension, the anti-inflammatory effect is gone, instead there is a pro-inflammatory response in those immune cells before even hypertension develops, and then an excessively large population of the CD161 splenocytes at least is present in the neonatal state and increases with age. So again, innate immune system in genetic hypertension is abnormally regulated by the autonomic system to trigger pro-inflammatory responses to endogenous antigen, and these induce significant pathologic damage and sustained hypertensive state. And here are all the people who helped us understand all of that, including several powerful immunologists without whom I wouldn't be able to interpret any of this stuff. And thank you very much.